Thank you for having us. This is Sheikh Daniel Anwar. Before anything else, I must say listening is the key to success, and it takes more than 1 hour of preparation for each minute of presentation time. Therefore, I request from all the listeners to listen carefully because good listener is one day a good speaker. According to Confucius, the man who moves a mountain begins by carrying away small stones. So here is a small milestone we have achieved by completing our final project of ethics and social responsibility, which I am going to share with you in a moment.
Let's first discuss what is ethics and social responsibility.
Rules of behavior, based on idea about what is morally good and bad. On the other hand, social responsibility is an ethical theory in which individuals are accountable for fulfilling their civic duty.
Our perspective is that we do not think anyone is perfect. It is our responsibility to do the best we can do to change the things we do not like. I think that is the part of ethics and social responsibility, and everybody is going to do that in their own way. The way we have focused on is provided by our Honorable Professor Israr Khan Raja, to whom this project is submitted to. Honorable Professor, I am representing the group number 10 and the members are Khadija Amir, Mir Munisa Shuaib, Ariba Imran, Umaima Fatima Junejo, Maham Wabab, Ramin Batul, Sheikh Daniel Anwar, Faisal Iqbal Sharif, Kandil Bukhari and Maria Zahra.
So, our presentation, genome editing creates two perspectives, just like coin or paper having two or more different sides, which creates some ethical dilemmas. So we have covering these dilemmas under one ethical statement, which
Is it unethical to perform experiments on human genome?
With two options.
The first one is genome editing is unethical and must be stopped immediately. And second is genome editing benefits outweigh the risk and ethical issues. What is gene editing or genome editing as it also known, and how does it work? Let's start by talking about genes. These are sections of the long DNA molecules coiled up inside each cell of every living thing, from microorganisms and insects to plants and animals, including humans. Think of DNA as the instruction manual for an organism. In people, genes can influence characteristics such as your height and the color of your eyes. DNA is inherited from our parents. We have a combination of two sets of genes, one from each parent, brought together to produce a new set of instructions and reshuffled in each generation. But sometimes these instructions carry errors, and these can create problems. Faulty genes can cause serious illness. Humanity has a long history of figuring out ways to modify genes. We have been crossbreeding plants to make them better to eat for thousands of years. But with gene editing, we have made a great leap forward. This increasingly accurate new technology is faster to use, relatively simple and cheap compared with previous methods, and promises huge benefits. In a nutshell, it works by identifying then cutting pieces of DNA. One way of doing this uses a component known as CRISPR to point out the precise DNA sequence within the gene to be altered. Then an enzyme called Cust9 snips through the DNA, changing it or allowing it to be replaced by another stretch of DNA that is introduced at the same time. This can either replace a faulty gene with a healthy one or change a gene to make it behave differently. Now, Ariba and Faisal will continue the presentation from the issues faced in genome editing.
Thank you, Sheikh. As Daniel mentioned, there is a difference of perspectives of perceivers which generates issues. Therefore, issues faced in genome editing is mentioned. The first one is embryo gene alternated. The second, overpopulation. The third, religion corners. Number four, miscarriage, stillbirth. Number five, embryos for experiments. Number six, social class. Number 7, off targets. Number 8, designer babies. Number 9, bioweapons. And number 10, bioterrorism. Okay, let's discuss them one by one.
The first one is embryos gene altered.
It is all about modifying the genes of an embryo can have unintended consequences which could harm not only the child but also their future descent but without their consent as the alternated gene could be in their sperms or eggs. The second one is overpopulation.
Humans by the millions falling ill and dying prematurely are all perfectly natural. While living longer is already creating social problems today, what about the overpopulated Earth after scientists remove all the disease in it? It would change how we would need to approach population control. Disease is one of the most effective methods of population control for the human race. The next is religious concerns.
Anyone who believes in the concept of God or a supernatural creator will have some level of ethical corner about the idea of humans genetically editing future generations. We already see this disadvantage to some extent in the abortion debate because of the implications that the choices being made are tantamount of playing
The
God.
next is miscarriage.
Embryos are being created simply for the sake of research. Experimenting in little life still in the womb could lead to the complications, including miscarriages, premature births, or even stillbirth, all of which are unthinkable. The next is embryos for experiment.
Many people have moral and religious objections to use of human embryos for research. Federal funds cannot be used for any research that creates or destroys embryos. In addition, NIH does not fund any use of gene editing in human embryos. The next is societal class. New technologies are always expensive. Only the households that could afford to take advantage of this service would perform gene editing on their children. That means the initial population surge from human gene editing would involve the wealthiest people. It would create a severe divide between those with money and the people without it. We could find ourselves living in a world where different classifications of genetic purity could lead to individualized approaches to healthcare, employment, and education. It would become the next step for those with wealth to safeguard their societal status.
The next is off targets. There always be the disadvantage of a mistake occurring when dealing with human genetic engineering. We are not a perfect people, no matter how smart the scientist happens to be. We have a general understanding of what our core makeup happens to be. But there is also the possibility that we do not have all of the pieces to the puzzles as of yet. Small changes to a growing baby could have a distorted effect that we might have unable to protect with our certain resources. Changing the genetic profile to remove disease might create miscarriages, birth defect, or a great risk of stillbirth. That's why a careful approach is to this subject is absolutely of necessary.
Designer baby.
Altering gene line to achieve beauty and intelligence will only further promote the idea of all races looking alike, decrease genetic diversity instead of expecting difference of people, different level of social class, different backgrounds, and turning everyone into a designer humans.
Bioterrorism. Like many technologies, genome editing can be used for both good and evil purposes. Biological war, also known as germ war, is the use of biological toxins or infectious agents such as bacteria, viruses, insects, and fungi with the intent to kill or incapacitate humans, animals, or plants as an act of war. Now I'm going to invite Kandil, Khadija, and Maham to accommodate the listeners with the benefits of genome editing.
Thank you, Faisal. As we all know, every piece of paper has different sides. Likewise, this topic also contains two perspectives. I'm going to cover the benefits of genome editing, which are as follow. First one is replacement for vaccines. Second, decrease infant mortality rate. Third, lifespan. Fourth, effect of editing genes on adults. Fifth, decrease infertility rate. Sixth, save endangered species. Seventh, allow for trait selection. Eighth, fight against cancer. Ninth, make xenotransplantation effective. 10th, eliminate inherent diseases.
Let's discuss them one by one.
The first one is replacement for vaccines. Using gene editing to create vaccines for viruses that previously did not have any is game changing. Research needs to continue as it can prevent the spread future epidemics and pandemics such as the one we are experiencing right now, that is coronavirus. Then the next is decrease infant mortality rate. There are several diseases that we can already detect during the fetal development process. Human genetic engineering can stop illness or disease before the child is born. Parents face a difficult choice when their doctor tells them that their child could face chronic pains, a lifetime of disabilities, or other health issues. Knowing that your baby would suffer for a few months and after that they will die, you are taking a gamble on their quality of life. The technology behind the engineering process could eliminate this issue, virtually guaranteeing that every child could be born healthy. Anything disease that comes from a potential carrier would slowly work its way out of the human experience.
The next is extend lifespan. It usually takes multiple generations to generate evolutionary movement within a species. As the environment changes, so must the physical traits of humans. Genetic engineering gives us a faster path forward that we can use to ensure the survival of our species. Altering our cells through this practice and make them more resilient to the natural aging process could extend our lifespan to levels previously thought to be impossible. We can feel better as we age too if we take care of our bodies, especially if we can program resilience against particularly dangerous diseases. Next is the effects of editing genes on adults. The procedure, if done on adults, not embryos, will not only affect their genes and it will not be passed to their offsprings. So basically, they have consent in whether they want their genes to be altered or not, and it will not affect their future generations either, which is ethically right.
Then the next is decreased infertility issues. Human genetic engineering could also make it possible for couples to have children when they would struggle at it naturally. We wouldn't need to worry about infertility because any sperm and egg combination could be edited to match the profile of the parents. This technology would make it possible to maintain population levels without going overboard on the family size. It is a necessary advantage to consider since the average fertility rate in the top 30 developed countries has declined by 50% in just 60 years. If a country does not have a birth rate of 2.1 per family, then the population shrinkage occurs. At 1.7, the United Kingdom is already there.
The next point we have is save endangered species. The same technology used to edit human genes can be used on animals. This could mean protecting a species like the Tasmanian devil, now endangered by an infectious cancer. It's everybody's planet, and there are huge implications for using this technology, a scientist says. I think scientists are interested in making sure their technologies or practices are rolled out in ways that are socially acceptable. Proposals to use editing as a wildlife conservation tool include applications that could help endangered species as well as the applications that could reduce or eliminate the invasive ones. Allow for trait selection. Most people concur that the primary benefit of human genetic engineering is to create a process where every baby is healthy at birth. Once we reach this goal, then what is the next step? It would likely be the selection of specific traits that future generations would want their children to have. You might get the opportunity to choose their hair color, eye color, or even gender. You would know how the child would look like at every age before they were born.
The next is fight against deadly disease cancer. New immunotherapy can be developed using genetic editing that can treat cancer, modification of T cells using CRISPR technology. It allows researchers to easily alter the DNA sequences and modify the gene function and locate and kill cancer cells. The next one is make xenotransplantation effective. One potential that CRISPR bring with its effectiveness is the application of xenotransplantation. In previous research trials, CRISPR demonstrated the ability to target and eliminate the endangered retrovirus, which reduces the risk of transmitting diseases and reduces the immune barriers. Eliminating these problems improve donor organ function, which bring this application closer to reality.
Eliminate inherited diseases. There are numerous diseases that require a genetic predisposition for their existence. That means a person risk for developing a cancer, Alzheimer's disease, and the other problems can be reduced because of this technology. When they exist on a single mutation, then we could eliminate them immediately before it impacts the development of baby system. Even smaller issues such as eczema could be resolved because of this proactive intervention. It could lead to treatment for people who are born without them as well. I predict we will abolish suffering throughout the living world. David Pierce once commented that our descendants will be eliminated by gradients of genetically pre-programmed well-being that are order of magnitude richer than today's peak experience. Although the topic is based on biological experiments, therefore, whether it is succeeded or failed, and it does not matter because failure is an opportunity to correct the errors. So now I'm going to invite Munisa, Maria, and Ramin to share success or failure story of genome editing.
Thank you, Kandil. We are going to explain some experiments of genome editing. This first one is He Jiankui. More than a year ago, the world was shocked by Chinese biophysicist He Jiankui's attempt to use CRISPR technology to modify human embryos and make them resistant to HIV, which led to the birth of twins Lulu and Nana. The team wanted to recreate this mutation using CRISPR on human embryos in a bid to render them resistant to HIV infection. But this did not go as planned. There are several ways they have failed.
The next is cure for HIV. For the first time, researchers have used CRISPR gene editing technology to try to treat a person infected with HIV. Scientists in China engineered human stem cells to mimic a rare form of natural immunity to the virus and transplanted them into a man with HIV and blood cancer. The gene-edited cells survived in the man's body for more than a year without causing detectable side effects, but the number of cells were not high enough to significantly reduce the amount of HIV in his blood. Then, the Berlin patient. In 2007, Timothy Ray Brown, initially known as the Berlin patient, underwent a bone marrow transplant to treat his leukemia. The bone marrow donor was special. However, in that he had a version of the CCR5 gene that confers immunity to HIV. Doctors hoped that the bone marrow transplant will replace Brown's HIV-susceptible blood cells with immune ones, and it did. After nearly 13 years, there is no sign of HIV in his blood, and his leukemia is in remission. In March, researchers reported that a second person underwent a similar procedure in Britain and was cured.
COVID-19 test. The Food and Drug Administration has authorized a COVID-19 test that uses the gene editing technology CRISPR and returned result in around an hour. Though the test was only authorized for emergency use, this marks the first time the FDA has allowed a CRISPR-based tool to be used in patients. CRISPR technology can quickly find and link into any genetic sequence in a specimen. The new test created by the biotech company Sherlock Biosciences uses one molecule to search for the virus gene in a patient sample. Then if the molecule finds the gene, it releases a signal that the system can detect. The standard method for COVID-19 testing, called PCR, also looks for tiny bits of the virus gene. However, that method is slow and takes specialized equipment to run. The recently approved Abbott test gives result in minutes but can only run on that company's platform. CRISPR-based technology, on the other hand, are relatively fast and only need basic equipment found in most labs.
I have always been suspicious of the assumption that great intelligence would be an unqualified benefit, that the madness that so often accompanies it can be cavalierly dismissed. So, I asked the question, suppose there were an entire subpopulation of extreme geniuses, well beyond anything that would occur naturally. What would that really looks like? For providing the answer of my question, I am gonna invite Umaima to compare ethics with the genome editing with the hope I and listeners get the answers.
Let's talk about ethics versus genome editing, shall we? Starting off, based on the ethical theory utilitarianism, we should be going with the choice that brings the most good to the people, which means clearly experimenting on embryos will not just bring good but genetic mutations as well to the embryo's offspring. Whereas performing the procedure on adults brings better and rather no bad at all, it involves their consent and no one else is harmed. The ethical decision of whether or not to go under genome editing is affected by one's own beliefs and faith in religion, one's own strength of integrity and personal values. So it solely depends person to person. Nextly, job availability and economic disparity is already an issue worldwide due to overpopulation. Most inequality in income and high rate of poverty can be expected. Gene editing sure can cure inherent diseases, but disease is nature's way of population control. Based on theory of justice, if gene editing is continued, the future generations will not be all treated equally and fairly. And also, due to unemployment, crime rates will increase as well. It is common ethics sense that purposely causing miscarriage or stillbirth cannot be justified. It is wrong in all sense of humanities. Nextly, due to high cost, this procedure will only be affordable by the high class of society and will increase already existing direct discrimination among the rich and poor. People with edited gene will be considered superior over those who are natural. This will result in extreme inequality among the societal classes. Consequences of this procedure are still unpredictable. Although it can cure cancer, the off targets can also cause cancer. Based on the theory of egoism, if the adult patient is warned about the off targets and the effects it can cause, then it depends on his own interest and betterment. I'd like to move on to the conclusion for our presentation. All of us, citizens, scientists, engineers, and future users of human enhancement methodologies, must proceed with conviction but also caution, with purpose but also extreme care. It's critical to appreciate the implications of the power of science, such as articulated by Richard Dawkins, that science is the most powerful way to do whatever it is you want to do. If you want to do good, it's the most powerful way of doing good. If you want to do evil, it's the most powerful way to do evil. Never before have we, or any other species on this planet, had such influence and so much power over the fundamental nature of our own biology. The non-therapeutic use of gene editing on human embryos was and remains unethical and illegal on every level. Yet now, we need to leverage attention on gene editing and human enhancement into a real conversation about the future of our species. A few areas of controversy concern whether there could ever be an agreement on what counts as safe enough to move from preclinical to clinical research, whether the rights of disabled people may be undermined, and whether human dignity would be significantly compromised by allowing germline genome editing. These areas of controversy have been addressed by references to the human rights framework, solidarity, notions of welfare, and social justice. Open questions remain concerning not only whether and so how clinical research involving human germline genome editing should be allowed, but especially concerning how to prevent increase in inequality, the discrimination of disabled people, the weakening of social justice and solidarity that may result from the diffusion of germline genome editing. These matters must be handled carefully and with extreme caution. As the saying goes, with great power there must also come great responsibility. And with that, I'd like to conclude our presentation of whether genome editing is ethical or not to an end. We'd like to thank you for taking out a few minutes from your precious time to listen to us today. We hope it was worth it. Thank you.